And the reason for this is because prior to the research I'm going to be talking about today, absolutely nothing was known about gene expression control at all. Absolutely nothing. So the experiments that I'm going to be talking about today, even though they are focused on expression of certain bacterial genes, and I know for the most part we don't care, but the reason why it's so important is because it really gives you an idea of the kind of effort it took to understand regulation of gene expression, even in the simplest systems, let alone our own. And so bacteria lend themselves to studying expression of genes and how that's regulated because it's a simple model system. Okay? None of this is in your book. It's a simple model system. It allows for you to understand how these kinds of things occur in the cell. Human cells or mammalian cells or eukaryotic cells in general are much more complex. And I'm going to talk about that a little bit at the end. But what I'm going to be talking about today was first actually started to be worked out soon after the DNA structure itself was established and determined. And again, bacteria lend themselves to these studies in a way that mammalian cells certainly never would. So that's how we know much about what we know. So I'm going to talk about overall metabolic control of how genes are regulated. Then I'm going to talk about in bacterial systems how they have their genes organized in structures called operons. Then I'm going to talk about two different types of operons. The operon that encodes genes involved in generating the amino acid tryptophan. Then I'm going to talk about an operon that is actually encoding genes involved in breaking down lactose to generate glucose for the bacterial cell use. And again, we really don't care how bacteria break down lactose. I know that. I'm not pretending. I also know that you really don't care how bacteria make their tryptophan. But again, it's a model system that set the stage for everything we know today. Okay. All right. So bacteria, like our own cells, bacteria have to conserve their resources. Why use resources to generate something that you don't need? And so there are two different mechanisms that not only bacteria use, but we use in our own cells. And one involves feedback inhibition. And we've seen this before when we talked about glycolysis and that wonderful enzyme phosphoglucokinase. So in generating tryptophan, the bacterial cells, if they have plenty of tryptophan around, there's no reason to generate enzymes and use resources to generate enzymes to make more. So the tryptophan itself functions in the form of feedback inhibition to bind to the first enzyme in its synthesis pathway to actually stop the pathway itself. And when the cell uses up its tryptophan, then what will happen is then there's no tryptophan around to inhibit the enzyme, and the pathway will continue on. However, there are other means of regulating gene expression, and that involves actually shutting down the genes that are generating these enzymes. And this is more for long-term conservation of resources. So the short-term conservation of resources will involve a feedback inhibition mechanism where the product of the pathway will bind to and inhibit one of the first enzymes acting in its own synthesis. The more long-term mechanism involves actually shutting down genes that are encoding the enzymes in the first place. Okay, and for a bacterial cell, long-term is 20 minutes. Okay, it doesn't take long. Most bacterial cells, their lifespan is basically 20 minutes. They undergo the cell cycle. They regularly undergo the cell cycle every 20 minutes or so, depending on the strain of bacteria. All right. So the first model system for studying gene expression we're going to look at is the operon that generates the enzymes involved in tryptophan biosynthesis. So an operon is actually a series of genes. It's a series of genes that generate proteins that form enzymes that all function in the same pathway. So I'm going to say this again. An operon is a series of genes, all arranged in the bacterial genome, one by one, and each gene in the operon will generate a protein that will become an enzyme, and all of these enzymes are going to function in the same pathway. Hence it's called an operon. So that's the way bacterial genomes are actually organized. They're organized so that all the genes necessary for a single pathway usually are all clustered together, which seems like it makes sense, right? Ours don't do that, but bacteria do. Very clever. And if we were to look at a bacterial chromosome, we would find the genes of an operon arranged as you see here. Before the genes of the operon, there are some regulatory sequences in the bacterial genome. One of them is an operator, and the other is a promoter. The operator is part of the promoter. The promoter region of the operon, or the promoter region of a gene, is where RNA polymerase will actually bind and begin to move down the DNA to generate RNA that will then be translated to make the enzymes necessary for the biochemical pathway. And in this case, it's tryptophan. What's also interesting, and this took a long time to work out, is that a distance away from the tryptophan operator, or the tryptophan operon, there's another gene that encodes what is called the tryptophan repressor. The tryptophan operon is interesting because it's called a repressible operon. It's a repressible operon, which means, and I have it up here, a repressible operon is an operon that normally is active. That means that the genes are usually so necessary for the bacteria to get by that most of the genes are constantly being transcribed. So a repressible operon is one that is usually active. It's expressing genes whose protein products are critical for the life of the cell. What I also have is a couple of other definitions for you. The operator sequence is a DNA regulatory element. It actually helps to control the operon expression. So RNA polymerase and its binding to the promoter and going through the genes is only part of the story. The operator sequence is also very important. And so when tryptophan is not around, the cell needs tryptophan, the bacterial cell needs tryptophan. It's a critical amino acid for generating its proteins. So therefore, when there's no tryptophan around, RNA polymerase will bind to the promoter. That RNA polymerase marches right through the operator sequence and begins to transcribe the genes in the tryptophan operon. Makes a single long messenger RNA off of that operon, and then the genes in that messenger RNA, the coding sequences in that messenger RNA, are translated by ribosomes, and we'll see that in a few slides from now. That's when tryptophan is absent. However, if in a bacterial system, the bacterial cell has plenty of tryptophan for its present needs, it will use tryptophan itself. It will use tryptophan itself to bind to this repressor protein. when there's enough tryptophan around, the tryptophan itself will be called a co-repressor. What it will do, it will bind to this regulatory protein, the repressor, and when tryptophan binds to this repressor protein, that repressor protein with tryptophan bound to it becomes a repressor molecule that binds to the operator. When that molecule binds to the operator, RNA polymerase is blocked and no RNA is made from the tryptophan operon. Isn't that clever? It took a long time, decades, to work out this system. And again, because it all involves bacterial genetics and mutations in the system, it took a long time to work this out. And again, this kind of pathway would have been, we are still working on similar pathways in mammalian cells, but it lends itself to looking at bacterial cells. Okay. Does anybody have a question, or can I repeat anything? Is this clear? You okay? Okay. All right. The next type of operon that was heavily studied, and the Nobel Prize was actually awarded for this work, which happened actually about 50 years ago, because at that point, like I said, absolutely nothing was known about how genes are expressed, let alone how it's controlled. And this was a series of French researchers over in Paris who did the work, and they were still working on this. It's still being studied because not everything is known. But nonetheless, what they studied was the lactose operon. They studied the lactose operon. Lactose is a disaccharide. It's glucose and milk sugar, the lactose, held together by a glycosidic linkage. That's lactose. And there's an operon that generates genes that are necessary for the bacterial cell to be able to use lactose. The lactose operon, in contrast to the tryptophan operon, which of course, tryptophan operon is involved in generating a critical amino acid, so it's usually active. In contrast, the lactose operon, they found, was usually inactive. So instead of being a repressible operon, the lactose operon is called an inducible operon. And it's induced by the molecule that needs to be metabolized. And in this case, that would be lactose. So the lactose operon consists of, it actually has a regulatory gene that encodes a repressor. Then the lactose operon has its promoter and operator sequence, and then the genes that make up the lactose operon. So this is the overall lactose operon. And when there's no lactose around, there's no point in the bacteria trying to use it. And so the lactose operon is off. And in this case, that regulatory gene that encodes the repressor protein will be on. That repressor protein automatically will bind to the operator and block RNA polymerase from coming through and transcribing the genes. Does that make sense? Right. However, when lactose is around, the bacteria say, well, what the heck? Let's use this. It's a great source of glucose. And so what happens in that case is the lactose itself actually functions as a inducer of expression of the operon. And in this case, what happens is the lactose, otherwise known as allolactose, it's an isomer. The allolactose binds to that repressor protein. Then that repressor protein becomes inactive. When the repressor protein is not around to bind to the operator sequence, then RNA polymerase automatically, when it binds to the promoter region, RNA polymerase will march right through that operator and then transcribe the genes necessary for lactose metabolism. The genes are beta-galactosidase, permease, and transacetylase. It's interesting because the beta-galactosidase, this is the enzyme that is going to cleave that glycosidic linkage between the glucose and the galactose. That's what this enzyme is going to do. The permease is an interesting enzyme. What this is a membrane protein. The permease is a membrane protein. None of this is in your book. So the permease is a membrane protein, and it functions as a secondary active transporter. In other words, it uses a hydrogen ion gradient outside of the bacterial cell. When it's bringing those hydrogen ions into the cell, it changes its configuration to be able to bind to lactose and bring it into the cell along with all of those hydrogen ions. So the permease is a transporter, and it's involved in secondary active transport to bring lactose into the cell. And then finally, we have the transacetylase. A transacetylase is, as this name would describe, actually is involved in transferring acetyl groups. It's an enzyme. Its function to this day, decades after much of this work was done, its function in this pathway is still not understood. How about that? For heaven's sakes. Anyway. So this is what happens with the lactose operon. When lactose is present, then it binds to its repressor. The repressor cannot bind to the operator. Therefore, RNA polymerase automatically can go through and transcribe the genes that are going to allow for lactose to be used by the bacterial cell. Okay. All right, does anybody have a question? It seems very clear when you look at it on the slide, but if you were actually doing these experiments, it's very, very complex. All right. Another thing that was identified while studying the lactose operon, there was another component to the gene regulatory mechanism that was identified, and that was the catabolite activator protein, or CAP. And what's interesting about the catabolite activator protein is it doesn't actually induce expression from the operon. What it does is it increases the rate of transcription. How about that? It increases the rate of transcription. And it works by actually binding to the promoter region of the lactose operon, and it binds to the promoter region of the lactose operon and opens up the DNA. So it's actually much more available to the RNA polymerase. And that's how it increases the rate of transcription. It binds to the promoter region of the lactose operon, opens up the DNA, so it's more accessible to the RNA polymerase, and that way the RNA polymerase will more easily bind and transcribe the operon. So how does the catabolite activator protein work? It actually works when there's lactose present, but there's not much glucose. So that means that the bacterial cell is going to want to break that lactose apart to get the glucose monosaccharides away and use it for its energy source. And in this case, it uses that famous second messenger, cyclic AMP. So cyclic AMP, if glucose is scarce, what happens is the cyclic AMP level in the bacterial cell is very high. And when cyclic AMP levels are high, they will be able to recognize the catabolite activator protein. Cyclic AMP will bind to it, cause it to become active, and when catabolite activator protein is bound to the cyclic AMP, it will then bind to the promoter and help to increase the rate of transcription of the lactose operon. If there's plenty of glucose present and available to the cell, if there's plenty of glucose available to the cell, then the lactose that's available isn't going to have to be used so much because the bacterial cell already has a quick and easy source of energy, plenty of glucose around. When there's a lot of glucose around, but because the bacterial cell really doesn't need much, doesn't need to break apart lactose, then there's not much cyclic AMP. That level is going to be low. Therefore, the metabolite activator protein will remain inactive. Then the RNA polymerase, which would ordinarily bind to the promoter and transcribe the operon, that polymerase is less likely to bind. And so the operon is only transcribed at a very, very, very low level. Okay? So it's not completely off, but it's transcribed at an extremely low level. And that's because the metabolite activator protein is not opening up the DNA of the promoter to allow for RNA polymerase to bind and transcribe the operon. Are we okay so far? Okay. And that's what we're going to study about bacterial gene regulation. But I want